Hello and welcome to this infection basics video. I'm Dr. Alid Roberts, and in this video we're going to look at some specific groups of exotoxins that cause membrane disruption in host cells, and then work through this with a key example in the form of the Staphylococcus aureus alpha toxin. And so one of the first things we need to ask ourselves are, what are membrane disrupting toxins? Well, in bacteria, they are a special type of exotoxin, meaning they are proteinaceous and secreted from the producing bacterial cell. Now, membrane disrupting toxins are a unique group of toxins that specifically target host cell membranes and compromise their integrity, interfering with normal cell physiology. And the cell membrane is essential for a number of reasons, not least because it separates the relatively uncontrollable external environment from the tightly regulated internal cytoplasm. And so when this is compromised, the consequences can often be fatal to the cell. Now, there are two main mechanisms by which these types of toxins achieve membrane disruption. The first is through the creation of membrane pores, where individual toxin molecules assemble into a multimeric complex that inserts itself into the host cell membrane. This complex spans the lipid bilayer, forming a discrete pore that disrupts membrane integrity, collapses ion gradients, and interferes with essential cellular processes, ultimately limiting cell viability. And a well-studied example of a pore-forming exotoxin is the alpha toxin from Staphylococcus aureus, which forms pores in red blood cell membranes, leading to hemolysis and the release of nutrients such as iron that support bacterial growth during infection. The second mechanism involves direct membrane damage, where toxins do not form defined pores, but instead destabilize the membrane structure itself. These toxins act enzymatically on membrane phospholipids, increasing overall membrane permeability, essentially making them a little leaky, which compromises cell integrity and can lead to cell lysis. And a good example of membrane-damaging exotoxins are phospholipase C from Clostridium perfringens, which hydrolyzes phospholipids in host cell membranes and plays a key role in tissue destruction and cell death during gas gangrene infections. Now, one of the best examples of a membrane disrupting toxin is that of the alpha toxin produced by the opportunistic pathogen Staphylococcus aureus. Now, as we've just mentioned, this exotoxin damages host cell membranes by forming pores, leading to the lysis of various cell types, including red blood cells, and contributing to tissue damage during infection. Now, during Staphylococcus aureus infection, the production of this toxin is tightly regulated through the AGR quorum sensing system, ensuring toxin production only occurs when bacteria are present to maximize its impact on the host. And at a genetic level, alpha toxin is encoded by the HLA gene, which directs the synthesis of the HLA protein. Now, this protein is also referred to as hemolysin alpha, reflecting its ability to lyse red blood cells. And so in the laboratory, the activity of alpha toxin can be visualized nicely using sheep blood agar. Strains of Staphylococcus aureus that produce alpha hemolysin generate clear zones of hemolysis surrounding bacterial colonies where red blood cells have been lysed, and so these zones of clearing show just how effective the hemolytic activity of the alpha toxin can be. Okay, so now let's look at how it achieves this damaging effect on cells. So the HLA monomer is synthesized in the cytoplasm of Staphylococcus aureus and secreted as a 33 kilodalton soluble protein. Now, in this monomeric form, the toxin is not biologically active. Activation requires the coordinated assembly of 7 subunits, which oligomerize to form a heptameric holotoxin, and so it is this assembled complex that constitutes the biologically active pore-forming structure of the alpha toxin. Now, the holotoxin has 3 distinct structural domains, each of which plays a specific role in pore formation. The cap domain forms the upper portion of the toxin and functions to stabilize the heptameric complex once assembly has occurred. Beneath this lies the rim domain, which mediates interactions with the host cell membrane, including binding to membrane lipids and host cell receptors. And these interactions are essential for correct positioning of the toxin prior to membrane insertion. And finally, we have the stem domain, which undergoes a major conformational change during pore formation. And that is because when these monomers are produced, the stem domain is actually folded away and only extends outwards as the oligomerization process happens. Now, a key thing about the alpha toxin structure is that it is rich in beta-pleated sheets, which allow beta strands from each subunit to align and form a beta barrel. And it's this beta barrel that spans the lipid bilayer of the target cell membrane, creating the membrane-spanning pore that underpins toxin activity. Now, with this structural framework in mind, we can now consider how the alpha toxin initially binds to target cells and assembles at the membrane, and there are two prevailing theories as to how this happens. The first is direct membrane binding. In this model, alpha toxin monomers interact directly with the lipid component of the host cell membrane, allowing them to assemble into the heptameric complex without the need for a specific protein receptor. Now, this mechanism has been shown experimentally as alpha toxins can bind to and form pores in artificial membranes that lack receptor proteins altogether. The second theory involves receptor-mediated binding. This is where alpha toxins have been shown to bind to host surface proteins, namely ADAM10, which stands for A-disintegrin and metalloproteinase 10, and the presence of this receptor significantly enhances toxin activity, and it's thought that these receptors make oligomerization and the formation of the holotoxin that little bit easier, and so cells expressing high levels of ADAM10 are more susceptible to alpha toxin mediated damage, indicating that receptor engagement increases the efficacy of oligomerization and pore formation. Now, in reality, these two mechanisms are not mutually exclusive. The alpha toxin can bind directly to membranes, but receptor-mediated interactions enhance targeting, efficiency, and toxicity, meaning that both processes likely contribute to pore formation during infection. So let's have a look at this in a visual way. And so we're going to focus more on the general mechanism. During Staphylococcus aureus infection, it will grow in number, it will quorum sense, and genes will be switched on, which includes genes associated with the alpha toxin. We then get the production and secretion of the toxin by the bacterial cell, and over time, this will start to accumulate within the external environment. Now, some of these monomers that we can see here will start to oligomerize. Now, this happens naturally, as we can see here, or perhaps, as we've previously discussed, it can happen in a more structured way in the presence of ADAM10 receptors, which increase the efficiency of this oligomerization process. Now, once the holotoxin has been fully assembled, the pre-stem residues unfold and extend outwards. This allows them to penetrate through the cell membrane of the target cell, creating the transmembrane pore, which has one of two key effects on the cell. First, it allows intracellular molecules to flow out of the host cell based on concentration gradients. Secondly, it allows extracellular molecules to flow from the environment into the cell, again dependent on concentration gradients. Now, some experiments have shown that calcium ions, potassium ions, adenosine triphosphate, as well as low molecular weight molecules between 1 and 4 kilodaltons are able to pass through the alpha toxin pore. However, it's the passage of water, mainly an influx of water, into the cell which is a problem. Because this influx is unregulated, the cells are unable to dispose of the excess water, and so they burst due to the increased osmotic pressure, leading to osmotic cytolysis. And that is essentially how the Staphylococcus aureus alpha toxin causes the lysis of targeted cells, making it a great example of a membrane disrupting exotoxin. And with that, we come to the end of this video. Hopefully you found the content useful, informative, and most importantly, easy to understand. Thanks for watching, and I hope you have a great day.